Where have you been? I've been waiting for you at the kelp forest. A few herds of noisy third graders came through, as well as a few college student types. They asked me some random questions from their marine bio assignments. We're not actually ready for the big tank just yet. We have to go back to the first half of the Hall of Fishes, but we'll be back here looking at the fishes in the big tank shortly. We'll start with a long, deep stare into the face of a big crab. A lobster would do just as well, and there's some large ones in the same tank as the moray. But the challenge is to get a nice look into the animal's face, specifically around the mouth area. If we had actually gotten to the crayfish in lab 2 weeks ago, we would have talked about the appendages of arthropods. Crayfish, lobsters, and this crab are all in the crustacea, but there are also the insecta, the insects, and the chelicerata, spiders, scorpions, and mites, and that sort of thing. These three form the largest clades within the Arthropoda, and they differ in the way that their segmental appendages specialize. We've talked about how homeotic changes generate complex body plans by taking what start out as similar repeated units of anatomy and then reshaping and repurposing some of the units into new structures. We talked in lecture about how it's evident that an insect's antennae and jaws are actually modified legs because of the way that mutations disabling the normal developmental sequence can result in structures similar to walking legs replacing the normal mouth parts and antennae, proboscipedia and antennapedia, respectively. In insects like the fruit fly, the appendages of the first segment of the head become the antennae, and then the appendages of the second segment become the mandibles, that is, the jaws. In crustaceans, it's very different, with the head region being comprised of a much larger number of segments, each of which contributes a pair of appendages to the workings of the head. Here in the crab, you've got antennae and antennules, both sensory in function, coming from the first two head segments. And then in the third segment, the mandibles, the crushing jaws. You can see these in my crab face here. Now behind and to the side of the mandibles, you can see little fan-like maxillae that assist in the circulation of water through the gill chamber, which is behind the head under the crab's carapace. These appendages have been modified to serve like exhaust fans, pulling water out from the chamber while fresh seawater is drawn in from the back of the carapace. Then, from segments behind the ones giving us the maxillae, you have feeding maxillipeds. These are the larger feeler-like things that help bring food to the mandibles. This means that there are between 7 and 11 appendages, from 7 to 11 head segments, and this is all before you even get to the periopods, or walking legs, which are the appendages of the thorax region of the crustacean's body. We almost never get to see this properly either in lab, because the crayfish are kind of small, or at the birch, because while the crabs and lobsters are definitely large enough to see things as beautifully as this, they rarely accommodate us by sitting with their face up to the glass. But since I have snippets of footage taken randomly and with no foresight, but spanning many semesters, here it is, just for you. Another bit of select footage is the moray, here a California moray, doing its eel swimming or anguilliform swimming. Unlike the wolf eels we saw earlier, morainids, that is, fishes in the family Morainidae, are real eels, or at least legitimately classified in the Anguilliformes of the Elopomorpha, that clade of relatively basal neopterygians that we talked about earlier. The morays at the birch are generally pretty chill. These are pretty big fishes, very happy just to hang out in their small tank because they live naturally in pretty small spaces anyways, whatever mini caves and crevices they can find on rocky bottom habitats. They don't usually come out of their caves to show us how they swim. At this point, some student unfailingly asks about the moray's pharyngeal jaws, an extra pair of jaws that pop out from the fish's throat once the big main jaws have grabbed a large prey item. These pharyngeal jaws, which are also equipped with sharp teeth, basically grab the prey and pull it into the pharynx towards the esophagus. Now, there are some harder but more interesting questions that I can't answer off the top of my head, but I'm more than happy to point them out to you. They have to do with the developmental origins of these pharyngeal jaws. Do they also start out as components of the embryonic pharyngeal arches? Now remember that the animal's main jaws started out as pharyngeal arches that were induced to differentiate into jaws as a result of the activity of neural crest cells, as well as other developmental signals. So maybe these pharyngeal jaws are also developing through a similar mechanism, maybe similar signals acting on a different pharyngeal arch. What do you think? I'll leave this question for you to answer. Next tank, to the left of the morays and lobsters, we have a bunch of senorita fish in the family Labridae, or wrasses. Pectoral fin swimming is clearly evident in these fishes, much like what we saw earlier with the surf perches. An advantage of using fin swimming rather than body swimming is maneuverability in tight spaces. Wrasses generally live in rocky habitats like the one seen in this tank, or in coral reefs, basically getting a lot of their food by picking things off of a hard substrate. They tend to have pointy fronts with small mouths and powerful teeth and jaws, capable of breaking shells. And their fin swimming allows them to move forward and backward delicately while facing a hard surface encrusted with shelly food items. Another thing that always comes up as we talk about the senoritas and wrasses generally is their mode of sex determination. As a human, you should be familiar with XY chromosomal sex determination, which is commonly seen across many animal taxa. But some animals, like birds and butterflies, use ZW chromosomal sex determination, in which males are the homogametic sex with two Z chromosomes, while females are the heterogametic sex, ZW. The W chromosome is like our Y chromosome, except that its presence causes development as a female. Still, other animals like turtles and crocodiles are famously subject to environmental sex determination, where the temperature of the nest during one particular point in development is what determines if the turtle or croc baby wears a pink bow or a blue one. As a side note, one mechanism of extinction that could precipitate from rapid climate change is with turtle and croc species dying out because all of the babies born will be of whatever sex is caused by the warmer temperature. Now, in the case of wrasses, which is actually another very well-known example, the fish are sequential hermaphrodites. These guys start out life as female and will undergo a spontaneous sex change under the right conditions. Many wrasses have social structures with territorial males that fiercely defend their space and all of the females living within it against other males. A female wrasse living as part of a male's harem does not undergo any sex transition unless there's a vacancy in the top management. If the male gets eaten by a seal or caught by an angler, a few of the larger females will change sexes. And then I guess they duke it out to see who gets to replace the previous alpha male. In the case of the California sheephead, which you'll see in the large tank up ahead, this goes along with a change in coloration from all pink as a female to a multicolored male with a black head, white jaw, red shirt, and black pants. Here we're looking at shark swimming. This was covered in the pre-Birch video. The prototypical swimming form of sharks includes a heterocircle tail with a vertebrae and body musculature extending up into the top lobe, a flat ventral surface broadened by extended pectoral fins, and the fish planing upwards when it swims and slowly sinking due to its negative buoyancy when it stops. In the case of the leopard shark or the smoothhound shark, the long, thin body almost undulates in an anguilliform manner, but unlike an eel, the frontmost part of the shark is not flexing, and so this is a subcarangiform body movement. While we're on the topic of sharks, the other main kind of swimming seen in elasmobranchs, This is another term used by marine biotypes to refer specifically to sharks and rays. The other kind of swimming seen in rays is the ragiform swimming seen here. It's basically underwater flight using large pectoral fins as wings. Thing about rays, though, is that they, like all chondrichthyes, have negative buoyancy, and they have their mouths at the bottom. So if they were to settle onto a soft bottom, where they live anyways, because often they feed on invertebrates living in the mud, they would not be able to take up water to breathe through the mouth, like we saw earlier in the boccaccio. The rays you see sitting on the bottom of the tanks at the aquarium will be showing you their water intake apparatus, the spiracles we talked about in the pre-Birch video. Water comes in through the spiracles into the bucopharyngeal space. That's the connected mouth cavity and throat. And then it contracts muscles, causing pressure to increase and causing the valves on the spiracles to close and pushing water out across the gill openings, which are on the ventral side of the ray. Some sharks also have spiracles. If you're able to get a close-up view of a leopard shark, you can see a small opening behind the eyes, but clearly a tiny hole like this would not be of much use to bring in all the water needed to meet the animal's gas exchange needs. But everybody knows, you say, that sharks require continual forward swimming to breathe, and if they stop swimming, they suffocate. That's ram ventilation, of course. Swimming forward with the mouth open will result in a pressure gradient pushing O2-rich water across the gill surfaces. Well, this is true for some pelagic sharks. A great white or a mako would asphyxiate if prevented from swimming. But evidently this is not true for leopard sharks. This guy is using active pumping of water in through the mouth and out through the gills, just like the scorpionfish and rockfish that are to its sides. By the way, the three fish species you see here are the spotted scorpionfish on the left, sometimes this is called a sculpin by local anglers, the leopard shark in the middle, and a gopher rockfish on the right. Of these three, only the gopher rockfish has a swim bladder. Why do the sculpin and the shark lack a swim bladder? Now, each will have its own reason. That was pretty easy. Now, if the gopher rockfish has a swim bladder, what's it doing on the bottom? Okay, now that we're looking at the big kelp forest tank with its adequate space to keep some of the larger open water swimmers happy, this would be a good time to talk about subcarangiform versus carangiform and thunniform swimming. All three are body swimming, as opposed to the fin swimming we've seen so far in the wrasses and surf perches, and the seahorse. But the differences between them are actually pretty easy to sort out. Yes, there is the question of what fraction of the body length is actually being swished back and forth. Subcarangiform swimmers use most of their body length in a typical tail beat, less for a carangiform and least for a thunniform. But this is kind of difficult to ascertain just by watching the fish casually swimming in a tank. Now, a really easy way of distinguishing subcarangiform swimming from the other two is by looking at the tail and caudal peduncle. Both carangiform and thunniform swimmers have narrow cylindrical caudal peduncles and a stiff sickle-shaped caudal fin. So if you were to catch one of these fishes, you could grab it by the caudal peduncle and lift it off the deck of the boat, and the stiffness of the tail would keep it from slipping through. If you tried doing this with a subcarangiform swimmer, the fan-like tail would just collapse as it slipped through your slimy fingers. So now that subcarangiform fishes are out of the way, we can talk about the differences between carangiform and thunniform swimmers. And here, I'll have to cheat for a second time. There are usually no thunniform swimmers at the Birch, so I'm showing you this from a different aquarium. I don't remember which one. It's a tuna. Now, comparing this fish with some of the similar sized jacks at the Birch, there's a difference in body shape. While both are very streamlined, the tuna is definitely thicker, and consequently not so able to flex its body during a tail stroke, at least not in the same way that the longer yellowtail can. Thunniforms differ from carangiforms in the location of the dark, slow twitch muscle that's used to generate the tail beats that propel the fish forward and sustain swimming. In the tunas, the dark muscle is medial, close to the vertebrae, whereas with a carangiform swimmer like the yellowtail, it's distal, that is, along the outside, just like where it was in the mackerel and the rockfish that we dissected last week. Okay, now here's a little quiz for you. What kind of body swimming is demonstrated by this fish? What about by this fish? Did you say subcarangiform for the half moon and carangiform for the golden jack? Good. Here's an easy one, even if they don't live in crevices. These are garden eels, and from the body shape alone, you can infer that they swim as anguilliforms. But do they classify into the same basal clade as true eels, or is this just an eel-like derived morphology from some other mostly non-eely clade? I'll leave it to you to do that bit of research on your own. Close by the large kelp forest tank, there's a nursery area where they have small critters, mostly babies, on display. Usually there's at least one shark egg case that's backlit, and you can see the baby shark kicking on the inside. This one is a swell shark's egg case, sometimes called a mermaid's purse, though you should be aware that mermaids don't really exist, and even if they did, I doubt they would need handbags. So from here to the end of the Hall of Fishes, we're looking at tropical collections, with corals and even giant clams forming the backdrop to a large variety of fishes. Now, I'm not going to be concerned with keeping track of all the species here, but we will be isolating on a few fishes to check out their swimming method. Let's see if you can tell. Start with this one. If I tell you that it's a blue bird wrasse, you shouldn't be surprised because its use of pectoral fin swimming is just like what we saw for the senorita fish earlier. Wrasses just swim like this, and apart from the bizarrely long snout, the body shape of this fish is the same as the other wrasses, of which there are many species in these tropical tanks. Now how about this one? Yeah, it's a puffer. Actually, a porcupine fish, because most puffers don't have the spines. But never mind any other aspect of this fish except for how it's swimming. Which fins are active? So now anytime you see continual activity like that, you can be sure that the muscles powering that activity are going to be dark in color. Why? Where would you expect to see the dark muscle in the wrasse, like the bird wrasse we were looking at just before? How about in this fish? This is a triggerfish, which is in the same order as the porcupine fishes and puffers, the Tetraodontiformes, as indicated in the handout. This is a highly derived order of acanthamorphs. Again, you don't see the easily identifiable traits. Most fishes in this order are reliant on fin swimming. But this fish is different from the porcupine fish, right? How do you describe the differences between the two? Now here's a blue tang or surgeonfish. Yes, it's dory. What kind of swimming are you seeing here? Pectoral fin swimming. Okay. Does this mean it's a wrasse? No, it's not a wrasse. Go ahead and Google up what family it belongs to. Here's something really neat. This is a sea robin. In last week's lab, We used the Miller and Lee key and moved right past the description where it said the fish had to have the lowermost rays of the pectoral fin detached from the fin. This is what the fish would look like if that were true. Look at how the fish uses these detached fin rays like legs to walk across the bottom. Okay, here's another one for you to ponder and make some commentary about. And here's the next one. And these shrimp fishes are really pretty bizarre. What can you determine from these fishes' behavior? Okay, now let's look at some cephalopods. This guy should look vaguely familiar. Squid? No, it's a cuttlefish. Also in the Decapodiformes, and essentially like a squid in many ways, except that it's more massive and it has a heavier, more calcified pen, the cuttlebone. These are also the ones whose ink is an important flavoring and coloring in coastal Italy and Spain. If you've ever had squid ink pasta or arroz negro, it's not really squid ink. And the Italian term nero di seppia, black of the cuttlefish, is taxonomically more accurate. This is also a cuttlefish. In fact, I showed you this one a couple of weeks ago during the squid dissection, didn't I? This is the flamboyant cuttlefish, as seen at the Birch. And here is its baby. This was in the nursery area near the swell shark egg case. And now here is the chambered nautilus, a coil-shelled cephalopod. Notice the absence of spire in the shell? It's shaped like what breakfast pastry? Here's the phylogeny quiz question for you. Is the chambered nautilus more closely related to octopuses or to cuttlefish? Just to the left of the nautilus and right before you exit the Hall of Fishes, there's this tank that looks like it's just full of rocks. But then on closer inspection, hey, that rock is actually a fish. And it's not just one, there are 4 or 5 of these stonefishes in the tank. And it's actually pretty scary to know how hard they are to see. Stonefish are the most venomous fishes in the world. People step on them when they wade in shallow water, and they get their feet or legs envenomated by the fish's fin spines, and then within an hour they're dead. You have to seek medical treatment quickly for the stonefish, and any sane Queenslander will have a good measure of respect for these things. Once out from the Hall of Fishes, students usually head outside to see the overlook of the Pacific Ocean and Scripps Pier, and to look and touch the animals in the simulated tide pools. It's really nice, but nothing that merits inclusion in this video. And then on the other side of the building from the Hall of Fishes, there's a featured exhibit that changes from time to time. Right now, the rotating exhibit centers on the Scripps collection of preserved specimens, dead things pickled in jars of preservative, with an emphasis on weird organisms. It actually looks like something I could develop into a core part of this aquarium activity, but I haven't gotten around to this yet. The last part of the visit takes us through the collection of mostly seahorses, but there are also pipefish and sea dragons. These sea dragons are the most awesome things you will see on the day that you visit the Birch. I can almost promise that.